Welcome to Matri Academy. Let's begin the chapter 19 Basic Techniques in Molecular Biology. Let's discuss the chapter Basic Technology in Molecular Biology. The first is DNA RNA extraction. During DNA isolation, C-tab is used for breaking cell membrane. ARS 2012 question. SDS provide overall negative charge to all the protein so that separation can be done only on the basis of molecular weight. During RNA isolation, DEPC is used to prevent RNA's contamination. Guanidine thiocyanose is used for RNA extraction. Question asked in IARI 2003. DNA fingerprinting. The term DNA fingerprinting had been coined by Alex Jeffrey in 1985. ARS 2012 question. In DNA fingerprinting studies, for the evaluation of high-order relationship, a similarity matrix can be calculated from band sharing data of each of the fingerprints according to the formula F is equal to 2m AB by m into A plus m into B, where m into A and m into B represents the total number of band in lanes A and B, and MAB is the number of bands which both lanes share. F is equal to 1 means pattern of bands are identical, and F is equal to 0 means no bands in common. A simple universal probe, a tandem repeat of GATA box has been developed from the sex chromosome of banded carrot. This probe is reportedly very useful in DNA fingerprinting of not only man but of many organisms including plants. DNA barcoding. Basically a type of DNA fingerprinting. In it, series of bands are used as a code for product. It is used to describe population at species and genus level. Little relevance within the species level. Circular DNA molecules, supercoiled or relaxed, show a much lower mobility in PFGE than do linear DNA molecules. This question asked recently in IARI JRF SRF 2025 exam. And also the question is in JRF 2025, that is the DNA barcoding. It is also known as DNA fingerprinting. Methylene bisacrylamide is used as a cross-linking agent to polymerize acrogel into polyacrylamide agarose gel. Agarose gel usually separates DNA fragments ranging from 0.3 to 23 kV size, while PASE polyacrylamide gel electrophoresis separates DNA fragments of 2 to 6 kb size. ARS 1997 question. An optical density of 1 at 260 nanometer corresponds to 50 microgram per ml of double stranded DNA or 40 microgram per ml of the single stranded DNA. DNA has maximum absorption at 260 nanometer. The ratio of optical density 260 to optical density 280 provides an estimate of purity of DNA. If it is 1.8, implies pure preparation of double stranded DNA. If it is greater than 1.8, means RNA impurities are there. If this ratio is less than 1.8, means protein impurities are there. DNA concentration of the sample is calculated as concentration of DNA per mg per ml is equal to optical density 260 into 15x dilution factor, and concentration of DNA in microgram per ml is equal to optical density 260 into 50x dilution factor by 1000. The probability for a particular tetranucleotide consisting of all the 4 bases to occur in a DNA molecule containing 50% AT and 50% GC base pair is 1 by 4 ka power 4. IARI 2003 question. What do you mean by CCC? It is a covalently closed circular DNA. Question asked in IARI 2003. Gel retardation technique. Gel retardation technique. On gel used to study nucleic acid protein interaction, linear double stranded DNA is mixed with protein and run in a gel. The complex of DNA protein will move slower as compared to normal DNA and covers comparatively a smaller distance, also known as band shift assay. Question asked in IARI 2004. When the probe is used to detect partially matched sequences, the hybridization temperature is reduced by 1 degree centigrade for every 1% sequence divergence between probe and the target. Sward and Cantor 1984 developed pulse field gel electrophoresis technique which resolved DNA molecules up to 1000 to 2000 kb in size. It uses alternatively pulsed perpendicularly orthogonally oriented electric field. Its disadvantage is that DNA sample do not run in a straight line track, which has been overcome and field inversion gel electrophoresis technique given by Carle et al. In 1986. Question asked in the IARI 2004. During SDS page, SDS provides a negative charge to all the protein so that their separation can be done on the basis of molecular weight only. Ethidium bromide is mixed in a gel since it binds with DNA and thus it fluoresces on the exposure of UV light and makes the DNA more visible. Ethidium bromide visibility is being measured by the orange color. Southern hybridization is used to identify DNA fragments from agarose gel. ARS Net 2001 question. Southern hybridization DNA, northern hybridization RNA and western blotting is used for protein identification. During southern blot analysis, a weight of 500 gram is placed on the filter papers. Southern hybridization technique is used to separate how much large size DNA fragment, that is up to 20 kilo base pair. ARS Net 2001 question. In filter hybridization with an oligonucleotide probe, the hybridization step is usually performed at 5 degrees centigrade below the melting temperature. Northern hybridization. It is used to identify RNA fragments. ARS Net 2001 question. The northern blotting involves RNA DNA hybridization. IARI 2003 question. In northern blotting, diisobenzoyloxymethyl paper and O-aminophenyl thioether cellulase paper are used. DMP paper is used in case of northern hybridization to separate and identify RNA fragments. ARS Net 2001 question. The nitrocellulose membrane is treated with proteinase K to remove proteins and to leave the denatured DNA bound to nitrocellulose membrane. ARS 2004 question. Western hybridization or Western blotting. It is used to identify proteins. ARS Net 2001 question. Labeled antibodies are used in case of Western blotting. IARI 2003 question. Antibodies used for Western blotting are generally labeled with iodine 125. MALDI-TOF MS, matrix assisted laser distortion ionization time of flight mass spectrometry, the full form of this. Chromosome landing was discovered by Tansley et al. In 1995. DNA footprinting is used to locate region of DNA not bound to proteins. ARS 2007 question. Nick translation technique. The process of Nick translation is widely used to label double-stranded DNA molecules, either radioactively labeled or non-radioactively labeled. Nick translation is catalyzed by the enzyme DNA polymerase 1 by its 5' to 3' polymerase activity and 5' to 3' exonuclease activity. Question asked in IARI 3 and 5. Alpha phosphorus 32 D-NTP is used in Nick translation, while in case of end labeling method, gamma 32 phosphorus DNTP is used. In radio labeling of DNA, DNA polymerase 1 label alpha phosphate group, while T4 polynucleotide kinase labels gamma phosphate group during end labeling of DNA molecules. IARI 2005 question. Probe used in hybridization is of DNA, RNA and proteins. Question asked in ICR JRF 2000. So these all are the short notes of molecular techniques used in biotechnology. I hope these all points help you utmost in your examination. So always like and subscribe my channels. Let's tune to my channel. Thank you for being here.